So thank you for that introduction. I'm going to be talking to you guys today about my project, Dr. Ed Sword's lab, which is characterizing the biofilm formation by the cystic fibrosis isolate Stenotrophomonas multifilia. So first I'll tell you guys a little bit about cystic fibrosis. This is an autosomal recessive disorder that results from a defect in the CFTR gene. This gene encodes for the cystic fibrosis transmembrane conductance regulator. It's a really big name that really just means decellular chloride channels. So when you have a defect in these, you see symptoms throughout the body, but within the lungs, you see iron dysregulation, which leads to thickening of the mucus. This then leads to immune cell infiltration, inflammation, and scarring. And all of these things together result in really an ideal environment for bacterial infections. Some of the really persistent colonizers include Pseudomonas aeruginosa and S. aureus, but there are also some less well-known organisms that are commonly isolated from cystic fibrosis patients. One of these is Stenotrophomonas multifilia. So this is a gram-negative bacillus. It can be found ubiquitously in soil and in water, but it can also be found around different hospital environments. So it's an opportunistic pathogen that has been implicated in a lot of nosocomial infections, most notably pneumonia and bacteremia resulting from respirator use. And I mentioned that this isn't one of the most prevalent cystic fibrosis isolates, so you might be wondering why we decided to study it at all. We've found evidence in the literature that points that this is maybe contributing more than we previously appreciated to disease. Some of the reasons we think this, first of all, it's been increasing in prevalence as an isolate from cystic fibrosis patients. We aren't sure right now if this is because infection rates are actually increasing or if this is because our detection methods have gotten better, but either way, we're really appreciating that this is found a lot more than we thought before. Researchers have also found that biofilm formation on bronchoepithelial cells can happen, and that's shown here. So this points to the fact that it might be persisting in this form within the lungs. Another group put this into a mouse and found that there was actually significant inflammation in the mouse model. This is important because right now we aren't sure if this is just in the lung because we've wiped out everything else, or if this is actually contributing to pulmonary exacerbations. This evidence really points to the fact that it might be a big contributor to disease progression. These strains are also really multi-drug resistant. So sequencing of the genome has shown that there are several multi-drug efflux pumps, but in addition, in my experience, I have yet to find a single antibiotic that has killed any of my clinical isolates in my lab. So when I started this project, I got a couple of clinical isolates from Dr. Bill Benjamin here at UAB. This included a lung isolate from the sputum of a cystic fibrosis patient and a blood isolate from a cystic fibrosis patient. We also got a mouth strain from ATCC. And so current literature in the field has indicated that bacteria are persisting within biofilms in the lung. And so I wanted to answer the question, does biofilm formation and composition differ depending on physiological niche of origin for each of these strains? So to do this, we did three kind of basic experiments. And the first was just to look at the overall growth rate of each of these. We then assessed overall biofilm formation, and then we wanted to look at the biofilm composition of these three different isolates. So to start with the growth kinetics, we just did this simply by taking optical density over time. These are the growth curves. And we found that there are actually some differences in growth rates between these isolates. And we saw that the sputum and the mouth isolate were pretty comparable, but that the blood strain grew at a faster rate. So this is something we just kind of kept in mind when designing our other experiments. So our conclusion here was that the growth rates between these strains did vary. We then looked at overall biofilm formation, and we quantified this using crystal violet assay. And the results of this assay were actually really surprising. So when I did this, I expected that the sputum strain, because it was coming from the lung, would be the best biofilm former, and that the blood strain, because bacteria traditionally are in a planktonic state within the blood, would be the worst biofilm former. And that is the opposite of what I found. So you can see that the sputum strain formed no detectable biofilm at any time point in vitro, and that the blood strain conversely formed the best biofilm at every time point. There might be some explanation as to why the blood made more than the mouth just based on those differential growth rates, but the sputum strain, there was no real explanation by growth rate as to why this didn't form any biofilm at all. So that's something we want to investigate in the future. So the conclusion of this was just that biofilm formation does vary by niche of origin. We then wanted to look at overall biofilm composition of these organisms, because this is something that hasn't actually been done in this strain. And there are three macromolecules that make up the majority of biofilm matrices for bacteria. This includes protein, extracellular DNA, and polysaccharides. So our strategy for determining which of these was making up the majority of the biofilm matrix was to systematically degrade each of them and then see which one conferred the biggest change in biomass. We did this by using DNase to degrade the extracellular DNA, proteinase K to degrade the protein, and then sodium metaferritate that opens saccharide rings to degrade the carbohydrate component. Just some details about exactly how this experiment happened. I normalized each of the strains to the same CFU per mil concentration. I then grew the biofilms for 24 hours because that's where all of the growth rates converged. I then washed the biofilms and added each degradation treatment for 2 hours, and then I washed these again and either stained with crystal violet or resuspended the biofilm and plated for viability. And moving forward from this point, I just did this with the mouth and the blood isolate because the sputum isolate didn't form any biofilm, and so I wouldn't be able to see any change after degradation. And then I also performed these experiments at 2 different atmospheric conditions, one with just ambient air and then also with added 5% CO2, because that was a little more representative of the lung environment. So after degrading with the DNase, we saw no significant change in either of the strains in overall biomass. So we concluded that extracellular DNA probably isn't playing a big role in these biofilm matrix. And this was confirmed by the viability plating where we didn't see any difference either. We then degraded with proteinase and again saw no change significant in either of the strains after the treatment. So we concluded that protein probably isn't playing the biggest role either. And this was again confirmed by the viability results. We then degraded with the sodium metaferritate and saw a really drastic difference in overall biomass after the degradation treatment in both of the strains at both atmospheric conditions. And so from this we concluded that carbohydrate is playing the biggest role in the overall biofilm matrix. This was confirmed with our viability results where we saw pretty dramatic decreases, although I don't have statistical significance on this one because I didn't have enough replicates. So the conclusion of this data was that the primary matrix component is carbohydrate. And so the overall findings of this research is, first of all, the ability to form biofilm depends on the site of isolation, although this is a very tentative conclusion. I only had one strain from each niche, so it's also possible that this is simply a strain difference, and we even need to test more to really hone in on that. But we feel pretty confident that carbohydrate is the major biofilm component of this organism because we saw such a drastic decrease in both of those strains from different places. And so moving forward, we would like to further identify what these matrix carbohydrates are. So we've narrowed this down to one macromolecule, but that's still a really broad category. So we want to figure out exactly which carbohydrates are involved, and then if we can identify these, then it's possible that we could use that to degrade the biofilm and maybe increase efficacy of antibiotics in vivo. We also want to look into why we saw absolutely no biofilm formation by that sputum isolate, because we think that this might be really interesting in its implications for cystic fibrosis. I have kind of 2 ideas about why that might be happening, and the first is that it might be that these strains have lost their initial attachment mechanism. So if they were already in a biofilm in vivo and then I got them, moving them to a new environment, they wouldn't be able to do that initial attachment and wouldn't be able to form biofilm. And the other option is that in cystic fibrosis there are already a lot of mucins present and there are biofilms formed by other organisms. So it could be that this is persisting within a matrix that's already provided for it rather than making its own. So that's something we want to look into further. With that, I would like to thank my lab, Swords Lab, and Dr. Bill Benjamin for providing the clinical isolates, and then also the Cystic Fibrosis Center for answering all of my questions on that part of my research. If anyone has any questions?
What kind of media did you use to grow the CF isolate?
It was
the biofilm.
It was NYG media, which is pretty complex. I am planning to use a minimal media and see if that makes any difference, because I know that's been.
And I would also try growing it in potentially modified CF sputum that you can get. And do you have any plans to do like dual species biofilms with other common CF isolates with the one you got from?
Yes, I am. So actually, one thing I didn't mention is that this particular strain was co-isolated with Pseudomonas. That's really common with this particular strain, and so right now I'm working on getting a GFP strain from a collaborator and trying to make an M-cherry strain of Pseudomonas and hopefully do those dual biofilms and really investigate that.
Do you have any plans to genotype your isolates and see if there's a genetic reason why they may display different biofilm formation?
Yeah, so I need to do MLST typing because that's something I haven't really done yet. There are some genes implicated, especially in biofilm formation. Those have been identified. So moving forward, we can get primers for that and really look into that.
What's your hypothesis for why this beard isn't from biofilms?
That is a really broad question that I have no data to back up yet. It really is those two things. So there has been data by another group that found that overall, cystic fibrosis isolates form less biofilm on average than non-cystic fibrosis isolates, which leads me to think that this maybe isn't a just my strain thing, that this is an overarching concept. And people have also shown that there is actually direct signaling between Pseudomonas and Stenotrophomonas, and that Stenotrophomonas can release a signal that changes overall biofilm structure of Pseudomonas. So that leads me to think that maybe it is some sort of interaction, but that's something we're going to look into.
Can you go back a couple slides where you were looking at the composition of your biofilm? I just wanted to see. Okay, so the CO2 concentration doesn't really change that.
Yeah, no, we didn't see much of a difference with that in any of the treatments. Any else? Thank you guys.